It's my pleasure to introduce our next guest from Forge Biologics, co-founder, president, and CEO, Dr. Tim Miller. As a reminder to attendees, if you'd like to ask a question of our speaker, please type it into the question box under the video player, and we will try to ask them on your behalf. The format for this fireside chat is around 25 minutes, and then if we have time near the end, we will try to ask those questions. So with that, let's get started. Tim, welcome. Thank you so much for being with us. To start, could you provide a couple minutes of introductory comments about Forge for investors new to the story?
Yeah, sure. Thanks for having me today, and thanks for allowing us to kind of share our story. It's a fun one, especially in this day and age, kind of coming out of the pandemic, and really as we start to see things in the market start to turn, and Forge is here as a somewhat novel, and definitely accelerating and advancing gene therapies. So Forge is a combined CDMO with a single product pipeline in clinical stage in phase 1-2. Companies at about 330 employees, and we are working with over 30 clients right now to manufacture AAV for gene therapy clinical trials. Based out of Columbus, Ohio, the company has over 20 GMP suites, and have made multiple 1,000-liter GMP runs for AAV. We also do plasma manufacturing, and love to share our story because we also get this really nice momentum behind it because of our own pipeline that a lot of the people in the company really enjoy hearing about and supporting. But it also allows us to come back and say, you know what, we get it, because we put our facility and our process and cell lines and stuff through the agency's review as well. So we've raised a Series A, B, and C to date, and look forward to sharing more.
Well, great. And did you have a video you wanted to show us?
We do have a video, as a matter of fact. It's a pretty short video, but we can go ahead and put that on. This gives a great fly-through of our facility. We've made a lot of changes. We founded the company in 2020 and closed the Series A mid-2020, so right in the middle of the pandemic, and started building out this facility. So flying through, that's our CTO, Dr. David Dismuke. One of our nice areas for gathering and holding events outside. We'll fly over to our loading dock to see our external backup generators, into our GMP warehouse, all the mechanical support to do HVAC. Nice because it's under roof, and you fly into our GMP warehouse. So today we have the facility's mostly full, about 80%, and with 1,100 pallet spaces. You can see we've gotten in front of a lot of the supply chain issues that I think a lot of other groups have. You fly into our freezer farm, and a little bit of movie magic as you fly out the back of this -20 into our PD and analytical space. And process development and analytical development are key areas many clients need as working with their AAV. And you hear a lot about scale up, which we have. You'll see some 5,000-liter bioreactors, and but we also have multiple 1,000-liter bioreactors actually at GMP. We also have the ability to scale down, which has certainly been helpful for many clients doing lead candidate selection. So all animal-free component media and process. We have a DMF on file with the FDA, and got the facility and the cell line and the process in front of both the FDA and the EMA. And you fly over to another part of the GMP facility, and you can see some of our suites. We do have the ability to do GMP filling, and as you zoom over here, you get to see the 5,000-liter bioreactors, so biggest of its class in the industry, and one of the first to be able to scale up to 5,000 liters. So fun time. And as we think about the future of gene therapy and genetic medicine, these types of bioreactors are really what's going to help bring gene therapy to the masses. And a great mural done by a local artist that shows the journey from discovery to injection. So fun for everyone to walk down on Main Street in the facility on this journey of hope.
Oh, great! That's a really nice video. So, what is the status of your build out? How far would you say you are towards, what, I guess, first, what do you have online, and how far are you in terms of complete build out and turning on suites and stuff?
Yep. So 20 GMP suites, multiple. I think we have 5 1,000-liter bioreactors, 2 5,000-liter bioreactors, a couple of 500s, a 200, and more 50-liter that I can count. So, we have the ability to manufacture for just about any scale, going through phase 1, 2, and into phase 3 trials. And we have about 25 analytical development assays, some of which have been, many of them are qualified. 1 or 2 have actually gone through the validation phase. So we can serve anything from eye programs to systemic, multiple indications. That's what the state of our facility.
Okay, well, let's dig deeper into some of your proprietary AAV design components, starting with your HAC 293 suspension cell line. Can you tell us what you think its key advantages are?
Yeah, probably the number one advantage. Well, there are 2 advantages. One is full traceability. A lot of us have suffered through those challenges when working with different organizations where the cell line isn't really commercializable. So we went back, we re-derived the cell line from its all the way back from a traceability standpoint. So have put the cell line in front of FDA, EMA, and others, so you can take the cell line full through. But more importantly, why would you do that? Well, the cell line, makes about 40% fulls without an enrichment step. So that's pretty high when the industry standard's 10 to 15. So, it's the case of, the better that you start with, the better you're going to come out with. So after enrichment, you're pushing 95% fulls. So those 2 things alone make the cell line, I think, really palatable. And a lot of clients come in to use that really at our research grade offering to kind of get into, their programs. So you can use it right out of the gate.
Okay, and how about advantages of your P-Ember helper plasmid?
Yeah, that's fun for us to talk about too. We have a molecular development department that focuses on trying to make improvements on cell lines in the process or the ad helpers. So we've shrunk down P-Ember a couple of times. We actually have some intellectual property and some publications coming out to talk about some novel findings there, particularly when thinking about how ad helpers help with AAV processing and manufacturing. But so clients can come in and use the cell line and the P-Ember plasmid to really increase their yields with stuff that's higher quality than average. So those 2 things alone are a reason why I think we bring a lot of clients to Forge. But the fun thing is that we're always trying to make things better. And so we have another version of the cell line and another version of the Ember plasmid coming out later this year that are actually even better. So look forward to sharing that data later on this year.
Okay, and then are you developing any advanced capsid designs, whether hybrid designs or some other approach?
We are. It's not a core function of the company, but we are always trying to find that next holy grail of AAV capsids that that can target tissue a little bit more explicitly or specifically with a little bit less side effects. So we do have 2 different ways that we're doing this, and happy to go on to more detail for those that are interested. But I guess suffice to say, yes, we are working on a capsid library, but again, not quite the core focus.
So here's a question that came up in one of our earlier discussions today. And I guess you could say it was maybe one-sided because the participants had their own internal manufacturing. But in terms of talking about CDMOs, while we've seen a lot of capacity come online, such as yourself, their point would be that, capacity doesn't equate to quality. So you're in a position to offer these bespoke solutions for clients. How would you push back on that argument that your capacity does come with quality?
Yeah, we read a lot of the same news as everybody else that's out there, right? And one of the things that if you watch our video and it's available online, you note that the facility is 200,000 square feet, with more than 20 GMP rooms and more to come. But the challenge with that is it's not just about building something, right? If you want to go out and build a 20, 25,000 square foot facility, it's about what you put in there, the quality systems that you put around it, the people and the process, right? So it's more encompassing than, a little more of a kind of simple build out. So, there are a lot of industry standard quality systems that are out there. When you start looking at your ERP, when you start looking at your LIMS, Forge has put that in place and has been in place for almost a year and a half. There's lots of news out there that you read about the difficulty about implementing, an ERP, and that's, I think, accurate. But again, it also helps with quality down the road because you're able to make sure that your COGS, your people, your resource planning has been effective.
So switching to the downstream side, what are you doing to improve the performance of your assays to characterize potency, purity, or other important product attributes?
Yeah, the agencies have really focused on potency assays as a key aspect of safety, and not just efficacy, but making sure that you can do that lot to lot to lot. We help a lot of clients go through their transition of often bridging work, going from an adherent method to a suspension method, for example. So, when you switch manufacturers or you switch manufacturing platforms, potency assays is going to be one of the ways that you can look at this to show that the product is comparable. The nice thing about it is because we have our own clinical program, we went through this with the FDA and the EMA. We did a lot of our own bridging work when we moved it to our product pipeline, facility, the cell line. So to have a good handle, I think, on what the agencies are looking for. We do potency assay develop on a kind of bespoke basis for clients that need it or are asking for it. Every group's a little bit different, but they all fall into two main categories, right, of biologic and non-biologic assays. So, we can help with that as well.
So you did mention, the fact that you develop your own gene therapy candidates, and what else do you think you're learning from that experience that allows you to be a better CDMO partner?
Our regulatory team under Chris Schilling, is able to really provide a lot of guidance. When we go to the FDA for a pre-IND or an IND submission, or we go to get scientific advice at the EMA, putting in our FBX 101 program, as one of the leads, has really been a differentiator for Forge. And while many clients may not say that it's in their selection criteria in an RFP, we do often get a lot of feedback that, hey, Forge gets it. Forge is one of us because you guys are really, you're putting your own product through the same things that we have to. So you get skin in the game, and that has really ended up making, I think, an important difference when being able to say, hey, look, our regulatory team can sit and talk with you or help you work through your pre-ID process for your CMC. That's, I think a lot of clients have really appreciated that help.
So, tying what you just said back to, the prior question about the assays, you understand from your own product how important it is to identify the appropriate product characteristics. In developing your assays, how difficult is it to specialize them to be able to identify, the key critical quality attributes that, at a minimum that would describe, what's there for potency to support the mechanism of action?
Yeah, the benefit of using the cell line, is really is that you can get into a platform process, right? And I think we all have, this vision, and I think one of the town halls is going on today as well with the FDA. But I think Peter Marks would say, look, we all have this vision of trying to expedite, particularly gene therapies for rare disease, in a safe and effective manner as possible. And there are groups out there that, are trying to use the same platform, the same process, the same cell line, the same vector, the same promoter, to be able to go and minimize the variability. So you try to get to this platform process, and you don't have to do this over and over and over again each time somebody wants to bring a program in. There's a lot of advantages for that because you can move faster, you understand your COGS better, the cost goes down a bit, you get economies of scale. So, that I think, is where, we're all really trying to get to.
Okay, and then just back to your own gene therapy candidate, 101 targeting Crab-A. Give us an update on where you're at there and what are the next steps for that program.
Yeah, we have a clinical update and a clinical presentation coming up at the American Society for Gene and Cell Therapy. And, to date, the data has been really, really promising. There's a couple of novel things about that program. it's a systemic gene therapy on top of or after a bone marrow transplant. So, notably when you go through a transplant process, the first thing that has to happen is, the initial immune system gets ablated. So you get a transplant and then a systemic AAV on top of that. The effect of that is you're essentially removing the immune system. So a lot of the immune challenges that I think some groups are worried about, you don't see that in our trial. And we've got a lot of the safety data and immune data to back that up. The interesting thing about it is that when you ablate the immune system and then give a systemic AAV, the AAV floats around inside the system for a bit longer. And that has the potential to change how we think about what a functional dose is. So, I think in our hands, a 1e to the 14th VGs per kg isn't 1e to the 14th anymore. It's more like 4e to the 14th, because you're getting a time component of how much dwell time the AAV is, in the body. So, as a result of both of those things, the trial data looks fantastic. The patients treated to date have had very little SAEs related to anything around the drug product, and really are seeing gross motor benefits, cognitive benefits, and definitely increased biological activity and a reduction of what's called cycosine, so the toxic substance that builds up. So we'll present a lot of this at ASGCT in a couple of weeks, and looking forward to chatting about it more.
Okay. As we move past the urgency of addressing the COVID-19 pandemic, what impact is this having on your business? Obviously, you kind of interact with the FDA as both a sponsor and a manufacturer. How has that changed your ability to interact with the FDA? You're trying to enroll your own patients in your own study. What's the impact there? And then finally, what's the impact in terms of supply chain constraints?
Well, I think that we're all very excited about seeing the initiatives coming out of the FDA right now about trying to, do accelerated approvals, particularly for, some of these disease populations, these patients. I think as we retrospectively look back on Forge over the past couple of years, it's been an interesting time because, many of the companies that were going to build out some level of manufacturing weren't able to, for, basically cash conservation, and they're coming to CDMOs to help. Forge, has been prepared for that, coming out of the pandemic. We didn't see a ton of supply chain issues. I mean, there was a couple of, I think, what we would think of as close calls. The only thing that we really see coming, any longer lasting COVID effects, if you forgive the pun, is really some of the microchip stuff, coming out of Asia. But overall, especially with the White House's, focus on biomanufacturing initiatives, we are really well positioned, I think, as an industry coming out of the pandemic.
And how about for your own clinical trial?
I think that there are more and more patients are more comfortable to go back to hospitals, right? And for us and for many other companies, it comes down to newborn screening. And, unfortunately, Krabbe was up in front of the RUSP again, and there's been a significant amount of news flow out there about it because the committee that basically gives the recommendation to HHS in a very odd way ended in a tie, which is against their charter and their bylaws. So we're really hoping for another shot at the RUSP because, again, as we come out of the pandemic, to the point of your question, there's more and more people going to the hospital to feel free that they can to seek care. And we're really hoping that newborn screening, particularly for Krabbe, really starts to take off from the aspect of being able to identify patients in a time frame where they can actually to have some level of effect for a gene therapy.
Okay. As you are ramping up your CDMO business, obviously tech transfer and customer integration are going to be critical to your success. Based on your initial customer experiences, what are you learning to do better?
Well, as a former developer, and many of the leadership team have been in gene and cell therapy for years and have been on the other side, we've experienced a lot of the challenges sometimes with working with a CDMO. So, working with our client development and client relations groups, it's really about open communication and constant communication when there are problems, and pretty open and transparent pricing. We try to do an almost all-in-one model where there's not a lot of hidden costs. So, things that, as we think about what we've learned, being more of consumer or a client to CDMOs, we try to make sure that a lot of the pain points that we've experienced, we don't, have for other clients as well.
Okay, and excuse me, no surprise to anybody. New capital's been more constrained for over a year now, so we see the earlier stage companies kind of making decisions to stretch their cash. Again, as you're ramping up tactically, in what ways have you adjusted your corporate strategy?
We, I think, saw over the past, 12 to, 18 months, that the pre-Series A companies or the companies that have been trying to raise Series A, that's been, I think, a challenge for them in the fundraising environment. We've come up with a couple of different models to help clients that are, having a little bit of trouble fundraising, and you can work through different, financial models. But I will say that, over the past probably 4 or 5 months, we've actually seen an uptick in a number of AAV clients in particular that have gotten funding. So there's a lot of dry powder out there for good science and good programs. And, I think we're seeing that turn actually really happen.
Okay. Maybe 2 forward-looking questions. One, we hear a lot of talk about AI these days. How do you think you might be able to integrate AI apps to improve the efficiency of your operations? And again, what I'm talking about here is not, utilizing a dedicated robotic arm or system, but, more of the ways where an app that, yet to be developed could improve efficiencies.
Well, 2, I think, comments to that. One, using AI sometimes can help work out some of the process, right? So if you're changing temperature, media formulations for a particular capsid, you can start to come up with an algorithm where you start to see, kind of going into it, if it's an overstep self-complementary capsid that's an AAV6, may have these media formulations or process that work better than a single-stranded AAV9 that's packaged well. So you can use some AI to help be predictive around that. But I mean, kind of to part of your point, we are actually putting in an automation lab, so those types of analytics come more readily to us, but also to our clients, so that they get to see data in a little bit more real time. So in being able to run, 24/7, an analytical or automation lab, it definitely has its place coming up in the next, within the next decade. So we're trying to get ahead of that as well.
Okay. And maybe a final question for you, Tim, and maybe a way to summarize a number of the topics that we covered here. But, how are you really trying to differentiate yourself to be the preferred AAV CDMO provider?
Well, we just announced yesterday, that we got QP declaration. So that puts us in a little bit more of a rarefied space, as having the ability in the United States to make a GMP AAV that can be used in clinical trials in Europe. So, that's one way to go about it. We also differentiate by a lot of our offerings, from the ad helper to the cell line, and the ability to take stuff all the way at Blaze, all the way through to commercial. All these things help differentiate Forge.
Okay, well, very good. Thank you for joining us for this session, and we'll also have Tim later on in our panel discussion for CDMO. So you should join us for that as well. So thanks so much, Tim.
Yeah, thanks for having me.